Welcome back to the deep dive. If the plasma membrane is the boundary of the cell, the place where life begins today, we are going on a deep dive into the complex world that exists just outside that boundary.
Mm,
We are exploring the extracellular space, drawing exclusively from Chapter 7 of one of the leading texts in cell and molecular biology.
that's our mission today. We often focus so much on what happens inside the cell, but the crucial insight from this source material is that the external environment, the materials, the structures, all those secreted proteins, it's not just passive packing material.
Right, it's not just filler.
Not at all. It plays a profoundly vital regulatory role. These external components are constantly signaling back to the cell, dictating its behavior, whether it grows or moves or differentiates or even just organizes into a functional tissue.
And to make sure we absorb the full complexity of this system, we're going to move systematically through this chapter structure, covering every major concept, every mechanism, and every visual aid presented.
Good idea.
If you can hold the image of human skin in your mind, the sources give us a perfect visual analogy for the two major ways cells interact with their environment.
Exactly. Let's look first at the epithelial tissue, which forms the outer layer, the epidermis.
Okay?
This is defined by cells that are really tightly packed and held together by specialized cell-to-cell contacts. The organizing principle here is, maximum adhesion and forming a solid boundary.
Now, if you move deeper into the skin, you hit the connective tissue, the dermis. This is structurally almost a mirror image, isn't it?
It really is. It's defined by a huge volume of extracellular material, or ECM, with just scattered cells like fibroblasts dispersed within it.
And that structural distinction, that's really the whole story here.
It highlights the central theme perfectly. In epithelial tissue, communication is cell to cell, but in connective tissue, those scattered fibroblasts rely entirely on specialized cell surface receptors to interact with the environment.
So that's the bridge.
That is the physical bridge, the literal connection point that allows the outside world to transmit messages inward. We are looking at structure and dynamic signaling all integrated into a single system.
Let's begin our journey right at the immediate cell surface, starting with the glycocalyx, or the cell coat. What exactly is this layer chemically, and why is it so important for a cell's immediate interactions?
The glycocalyx is essentially a carbohydrate cloud. It's a layer of oligosaccharide projections that extend outward from interval membrane proteins and certain membrane lipids, a concept introduced when discussing the plasma membrane itself.
Okay.
It's very prominent on cells that form barriers, like the epithelial cells lining our digestive tract, where it forms this thick, fuzzy coating, which you can see visualized in Figure 7.2b.
It sounds like the cell's immediate interactive armor. What specific functions are bundled into this layer?
Well, it has a few core roles. First, it mediates cell and cell substratum interactions. Second, it offers mechanical protection from abrasion. Third, it acts as a primary barrier to particles trying to reach the plasma membrane. And critically, fourth, it binds regulatory factors, like hormones or signaling molecules that act right on the cell surface, serving as an initial receptor site before those signals are passed inward.
Moving outward from that immediate coat, we encounter the extracellular matrix, ECM, of animal cells. We sometimes casually dismiss the ECM as glue, but the sources quickly correct that idea, emphasizing its role as an organized network.
Precisely. It's so much more than just padding. The ECM provides crucial physical and biochemical signals that actively regulate cell shape and activity.
And the sources cite a classic experiment that really demonstrates this active regulation, right?
Yes. If you take cultured cells, like cartilage or mammary gland cells, and you use enzymes to digest the surrounding ECM, the cells immediately decrease their specialized synthetic activity. They lose their differentiated state.
Stop doing your job, basically.
They do. But here is the remarkable finding, a concept that's revisited later in Figure 7.29. If you simply add the ECM components back to the dish, the cells restore their differentiated state and restart the synthesis of their specialized products.
Wow. So it's definitive proof that the environment is constantly sending these crucial molecular identity signals.
Absolutely. The outside structure dictates the inside function.
And structurally, how does this matrix differ from the materials inside the cell?
The key structural difference is architectural. Unlike the compact globular proteins found inside the cell, like enzymes, ECM proteins are characteristically extended, fibrous species. They are designed for self-assembly, capable of linking together to form the huge, stable, interconnected three-dimensional network we see modeled in Figure 7.5.
Now, within this general matrix, we find a highly specialized structure called the basement membrane, or basal lamina. This isn't loose fibers. They're a tight, highly defined, continuous sheet, though surprisingly thin, only 50 to 200 nanometers thick.
And its specific location underscores its importance as a foundational layer. It surrounds individual nerve, muscle, and fat cells. Crucially, it underlies all epithelia, like the skin epidermis we mentioned, and the inner lining of blood vessels.
So what's its function?
It has a dual role, really. It provides mechanical support and acts as a substratum for cell migration, while also separating tissues and maintaining those vital cell survival signals.
The sources highlight its vital role as a specialized barrier. Can you elaborate on the clinical implications tied to this barrier function, specifically regarding the kidney?
Certainly. In the kidney, specifically the glomerulus, which is shown in figure 7.4b, the blood is filtered under pressure through a double-layered basement membrane.
Okay.
This layer is an essential filter that prevents large blood proteins from escaping into the urine, and the clinical consequence of its failure is critical. In long-term diabetics, this glomerular basement membrane often thickens abnormally, which disrupts the filtration process and can lead directly to kidney failure.
And it's also a barrier to cancer.
Yes, it acts as a physical barrier against cancer cells, which must actively degrade this membrane using enzymes to invade new tissues.
Now let's drill down into the major molecular architects that build this system, starting with collagens. If the ECM is a complex structure, collagen is absolutely the insoluble high-tensile framework.
It is the ultimate structural component, found only in extracellular matrices. The sources highlight its almost unbelievable tensile strength. A 1 millimeter thick fiber can suspend a 10-kilogram weight.
That's incredible.
It is, and it's everywhere, constituting over 25% of all human protein, making it the most abundant protein we produce.
And while there are 27 distinct types, they all share that defining architecture, don't they? The 3 alpha chains wound into a unique rod-like triple helix, which we see in figure 7.6a.
Yes, and the stability of that helix depends entirely on meticulous post-translational modification. These alpha chains are rich in proline and lysine residues, which must be hydroxylated to form the necessary internal hydrogen bonds that stabilize the triple helix structure.
And this chemical requirement leads directly to one of the most famous clinical connections in biology, scurvy.
Exactly, the classic sailor's affliction.
So how does scurvy tie into collagen modification?
Scurvy results from vitamin C or ascorbic acid deficiency. Vitamin C is required as a coenzyme for the hydroxylating enzymes that modify proline and lysine.
So without vitamin C.
Without it, the collagen is synthesized improperly. It's structurally unstable. And this manifests as inflamed gums, poor wound healing, and brittle bones. It's a profound example of how a simple vitamin deficiency cripples the foundational strength of all connective tissue in the body.
Let's focus on the fibrillar collagens types 1, 2, and 3. How does their assembly achieve that massive tensile strength?
The key is organization and permanent cross-linking. These collagen molecules self-assemble, aligning side by side in a staggered fashion, which gives them maximum resistance to pulling forces. This staggered alignment produces a distinct, repetitive banding pattern, about 67 nanometers, that's easily seen in electron micrographs, like in figure 7.6b and c. Furthermore, they are strengthened by lifelong covalent cross-links between lysine and hydroxylysine residues.
And that continuing cross-linking is why tissues stiffen over time, accounting for the decreased elasticity in older skin and bones. The structure is literally hardening year after year.
That's right. And new research, shown in figure 7.610, even refines our understanding of the fiber structure, suggesting they are spirally twisted nano-sized ropes.
The way these fibrils are organized on a macro level dictates the functional properties of the tissue. In tendons, where pure unidirectional resistance to pulling is needed, the fibrils are aligned perfectly parallel to the long axis of tension.
Right, but if you look at the cornea, as in figure 7.7, which needs strength and transparency, the organization has to be different.
The cornea utilizes a brilliant architectural trick, a kind of biological plywood structure. Fibrils are organized in layers, but the fibrils in one layer are nearly perpendicular to those in the layer above and below. Why does this specific arrangement promote transparency?
Well, by using this highly ordered perpendicular structure, they minimize the scattering of light waves, which maintains optical clarity while still providing exceptional strength to the front of the eye.
It's a perfect example of architecture serving a dual function, strength and visibility.
Exactly.
We should also touch on the structural failures, the fibrillar collagen disorders.
These defects really highlight the importance of the correct structure. Fibrosis, for example, is the excessive pathological buildup of collagen in organs like the lungs or liver, leading to scarring and loss of function.
And on a genetic level.
Genetically, osteogenesis imperfecta, that's a type I mutation, causes brittle bones. Certain dwarfisms result from type 2 mutations, which alter cartilage. And Ehlers-Danlos syndromes involve defects that lead to hyperflexible joints and highly extensible skin.
We noted that not all collagen is fibrillar. Non-fibrillar collagen type 5 is restricted to the basement membrane. How does its structure differ, and why is that so important for its barrier function?
Type 5 collagen is architecturally distinct, as we can see in figure 7.8. Unlike the rigid rods of type I, type 5 has non-helical segments interspersed along its length, which provides flexibility and globular domains at the ends, which serve as interaction sites.
So it's built to be flexible, not rigid.
Precisely. This structure is perfectly suited to form a stable, flexible network or lattice, the foundational support required by the basement membrane, which is modeled in figure 7.12.
And again, a clinical link. Mutations in type 5 collagen are associated with Alport syndrome, an inherited kidney disease specifically caused by a disrupted glomerular basement membrane. It shows how the failure of this single architectural component compromises the entire kidney filter.
Right. And moving from the steel frame to the cushioning element, we have proteoglycans. These are the hydrated gel that fills the space between the collagen fibers.
These structures are immense, as you can see in figure 7.9a. A core protein is covalently attached to long linear chains of glycosaminoglycans, or GAGs. The GAGs, like chondroitin sulfate, are repeating disaccharides that are highly acidic due to abundant sulfate and carboxyl groups.
And the negative charge is the whole story. The sheer quantity of sulfate and carboxyl groups gives the GAGs a tremendous negative charge density.
I see.
Individual proteoglycans, particularly in cartilage, can link to a huge non-sulfated JAG called hyaluronic acid to form these giant complexes. The source notes these can reach a molecular mass of 3 million Daltons, occupying the volume of an entire bacterial cell.
Single complex that big with that much negative charge must attract an enormous amount of water.
Exactly. The high negative charge binds vast numbers of positive ions, which in turn draw in a large amount of water via osmosis. This forms a porous, hydrated gel that is surprisingly stiff. It is this osmotic swelling pressure that enables the matrix to resist strong crushing or compression forces.
So the collagen resists the pull, and the proteoglycan gel resists the crush. That's the essential structural teamwork in tissues like cartilage.
It is. In bone, this same gel structure is present, but it's hardened by calcium phosphate salts deposited throughout the matrix.
Next, we have the molecular bridge and trail marker, fibronectin.
Fibronectin is a fascinating example of modular biological construction, shown in figure 7.10a. It's a large protein composed of 2 polypeptides joined by disulfide bonds, built as a linear array of about 30 independently folding modules. It's, designed to be flexible and multi-purpose.
What are its 2 primary binding activities that make it such a crucial linker?
First, it binds to multiple ECM components, collagens, proteoglycans, and other fibronectins, literally weaving the stable, interconnected network together.
And it's dynamic.
Very. What's truly intriguing is that this binding activity is not static. It's dynamically enhanced by mechanical forces. When physical forces pull on the fibronectin, the modules can unfold, exposing previously hidden binding sites.
So the stronger the tissue is pulled, the stronger the fibronectin anchors become.
Yes. Second, and equally vital, it binds directly to cell surface receptors, anchoring the ECM to the cell itself. This is visually demonstrated in the chapter's opening micrograph, showing a cell spreading and adhering precisely where fibronectin is placed.
Its role in embryonic development is hugely significant, acting as a migratory trail marker. Can you expand on how cells physically use this protein to navigate the developing embryo?
Absolutely. Think of it as a set of molecular guide ropes. Migrating cells, such as neural crest cells, which have to venture great distances throughout the developing embryo to reach their final destinations, follow pathways that are dense with fibronectin fibrils.
Like a highway for cells.
Exactly. In culture, as shown in figure 7.10c, cells adhere exclusively to strips coated with fibronectin, demonstrating a strong preference and reliance on this molecule for guided movement.
It's also required for the shaping of tissue's morphogenesis.
That's right. For complex branching organs like the salivary gland, kidney, or lung, The formation of clefts is essential. If you inhibit fibronectin function using antibodies in a developing salivary gland, as in figure 7.10e, it completely prevents cleft formation and subsequent branching.
So it's actively guiding the shape of the tissue, not just providing passive support.
That's the key.
Finally, we must mention laminin, the cross-shaped glycoprotein foundational to the basement membrane.
Laminins are characterized by their distinctive cross shape, composed of 3 polypeptide chains linked by disulfide bonds. There are at least 15 types, and they are powerful influencers of cell migration, growth, and differentiation. Their importance is immediate and profound.
What is the classic example of laminin guiding cell movement?
The migration of primordial germ cells, which is shown in figure 7.11. These are the precursors of sperm and egg, and they begin their journey outside the developing embryo. They must traverse laminin-rich surfaces to reach the developing gonad.
So they're following the laminin path.
They possess specialized surface proteins that bind strongly to laminin subunits, using the network as a precise, required migratory path.
And functionally, laminin works with type V collagen to create the basement membrane scaffold, right?
Yes, as we see in the model in figure 7.12, they work in parallel. Laminin molecules and type 5 collagen molecules form separate but interconnected networks, essentially weaving together the flexible, strong foundation of the basal lamina. The vital nature of laminin is shown in developmental biology. Mouse embryos unable to synthesize laminin fail completely to form basement membranes and die upon implantation.
It's non-negotiable for tissue stability.
It is.
We've established that this matrix is highly structured, but the sources stress that it is also incredibly dynamic. Let's look at the dynamic properties of the ECM, specifically how it is constantly being remodeled.
The ECM is in a perpetual state of flux, stretched, contracted, degraded, and reconstructed. The agents of this change are the matrix metalloproteinases, or MMPs. These are a family of zinc-containing enzymes, either secreted or membrane-anchored, that collectively can digest nearly every component of the ECM.
Physiologically, this remodeling is essential for processes like wound healing, cell migration, and blood vessel formation. But pathologically, their excessive activity is disastrous.
Indeed. Excessive MMP activity is implicated everywhere. In arthritis, where they degrade joint cartilage, in gum disease, in the development of blood clots leading to heart attack, and most critically, in tumor progression.
Right. Cancer cells weaponize MMPs to literally carve a path through the basement membrane and connective tissue to initiate metastasis.
Yes, and that specific link to cancer metastasis is fascinating, but there's a huge insight in the sources regarding clinical attempts to stop this process.
This is a crucial takeaway for our listener.
It is. Early research showed that using MMP inhibitors in animal models effectively halted cancer spread. Based on this, there was a massive push to develop MMP inhibitors for late-stage human trials. However, they largely failed.
Why did targeting such a central enzyme fail so dramatically in humans?
The insight is complexity and redundancy. The tumor microenvironment is far more sophisticated than initially modeled. Cancer relies not just on one pathway for invasion, but dozens of redundant mechanisms.
So blocking one path wasn't enough.
Exactly. Simply inhibiting one enzyme, the MMP, was easily compensated for by other, less understood pathways, rendering the treatment ineffective in advanced human disease. It shows the incredible challenge of translating basic biological targets into clinical success.
We've spent a lot of time defining the environment. Now let's bring the cell back into the equation and examine the critical linkers. This takes us to the most important family of receptors for cell-to-ECM attachment, integrins.
Integrins are found only in animal cells, and they perfectly embody their namesake. They integrate the outside environment with the inside cellular machinery. They are the definition of a multi-purpose receptor.
Structurally, they are heterodimers, made of an alpha chain and a beta chain linked non-covalently. They look like a pair of legs connected to a globular head, as we see in figure 7.5, spanning the membrane with a small cytoplasmic domain.
And the function of integrins hinges entirely on a dramatic conformational switch, beautifully demonstrated in figure 7.13. Crystal structures showed them bent sharply at the knees.
The bent conformation in figure 7.13a.
Right, and the consensus is that this bent conformation is the inactive state low affinity, incapable of binding ligand.
So the switch to the upright conformation is the active state. And the most compelling part is how this switch is triggered, leading to inside-out signaling. An event inside the cell rapidly activates the external receptor. How does that signal transmit across the membrane?
The mechanism is specific and efficient. as shown in figure 7.12. A cytosolic protein, most famously talin, binds to the small cytoplasmic domain of the beta subunit. This binding physically forces the two cytoplasmic tails to separate.
A physical push.
A physical push that then propagates a conformational change all the way up through the transmembrane legs, forcing the molecule into the upright, high-affinity active conformation.
It's a structural lever. The cell pushes on the tails and the head flips open, ready to engage the matrix. This is critical for processes like blood clotting, where a platelet must only aggregate when danger is sensed internally.
And the signal also flows in reverse. Outside-in signaling. Ligand binding to the external head, say fibronectin binding, induces conformational changes in the cytoplasmic domain.
What are the consequences inside the cell when this outside-in signal is received?
The cytoplasmic changes alter how the integrin interacts with internal adapter proteins. This initiates a signal cascade that affects everything from the cytoskeleton, leading to changes in actin polymerization, to the nucleus, activating cytoplasmic protein kinases like FAK and REC, and potentially driving gene activation.
This signaling is literally life or death, isn't it?
It is. Normal cells have a dependency known as anchorage dependence. They require integrin ligand binding to transmit those life-saving signals. If they are put into suspension, they often die.
And cancer cells?
The frightening capability of malignant cells is that they often bypass this dependence, allowing them to survive when detached from the matrix during metastasis.
Let's discuss the common molecular language used by many integrin ligands.
The tripeptide motif, RGD. That's arginine glycine aspartic acid.
The RGD sequence is recognized by many integrins and is found ubiquitously in key ligands like fibronectin and laminin. It serves as a near-universal cellular recognition signature for the ECM.
And this knowledge has direct, life-saving therapeutic consequences in platelet aggregation. Yes, the alpha-I beta-3 integrin is highly expressed on platelets. When activated, it binds to soluble RGD-containing proteins, like fibrinogen, linking platelets together to form a blood clot. We can exploit the RGD sequence to prevent unwanted clotting.
So how exactly do these pharmaceutical analogs work?
By synthesizing small RGD peptides or analogs drugs like agrestat or integrillin or specific antibodies, these compounds competitively inhibit the binding site on the platelet integrin. They essentially fill the RGD slot before the natural clotting proteins can, preventing the platelets from linking together.
Preventing the inappropriate formation of blood clots that cause heart attacks and strokes.
Precisely.
Integrins connect the external world to the internal cytoskeleton via two specialized structures. First, focal adhesions. These are dynamic, scattered sites of attachment observed when cells like fibroblasts spread onto a culture dish.
The core structure here is the link to the actin cytoskeleton. Focal adhesions are dense clusters of activated integrins whose cytoplasmic tails connect via adapter proteins like talin and vinculin directly to bundles of actin filaments.
We've established they are sensory structures, but they're also motor structures involved in locomotion and generating traction.
That's because they contain contractile proteins, actin and myosin. They aren't just anchors, they are motors. Figure 7.18 visually confirms this, showing a cell actively deforming a substratum and deflecting tiny posts. The cell is pulling on its environment, actively generating tension and traction forces essential for movement.
This pulling action brings us to one of the most exciting concepts in modern cell biology: mechanotransduction, converting mechanical forces into biochemical signals. How does a cell feel the stiffness of its environment?
The physical force itself generates the signal. For example, pulling on fibronectin-coated beads activates Sokuhara kinase inside the cell. The prevailing hypothesis suggests that the conformational changes in those adapter proteins, like talin, caused by the physical stretching force, expose new binding sites, which then activate the signaling cascade.
So the physical state dictates its biochemical activity.
Yes. And the power of this mechanotransduction is nowhere more evident than in the case of stem cell differentiation, which the sources highlight as a massive insight.
This is truly revolutionary.
It is. Mesenchymal stem cells, the precursors to various cell types, can be made to differentiate into completely different cell lineages: nerve cells, muscle cells, or bone cells, based only on the stiffness or elasticity of the substrate they are grown on.
Just the stiff.
Just the stiffness. A soft substrate induces nerve differentiation, a slightly stiffer substrate induces muscle, and a very stiff one induces bone. The focal adhesions act as the sensors, reading the physical environment and transmitting that critical instruction inward to determine the cell's genetic destiny.
Now let's contrast that dynamic actin-linked anchor with the second major structure, hemidesmosomes. These are the tightly stable attachments linking epithelial cells to the underlying basement membrane.
The structural difference is paramount. Hemidesmosomes utilize a dense cytoplasmic plaque, but the filaments coursing outward into the cytoplasm are much thicker and consist of keratin, which are intermediate filaments.
Ah, so not actin.
Not actin. Unlike the transient contractile actin of focal adhesions, intermediate filaments provide stable, supportive tensile strength and structural continuity.
So the cell uses two fundamentally different cytoskeletal systems for attachment. Dynamic actin for movement and mechanosensing, and stable, load-bearing keratin for static anchorage.
Precisely. The hemidesmosome uses the specialized alpha-6 beta-4 integrin to link the keratin intermediate filaments via the plaque to anchoring filaments in the basement membrane.
And again, when this stable anchorage fails, the clinical consequences are severe.
They are devastating. Bullous pemphigoid is an autoimmune disorder where the patient produces antibodies that attack specific hemidesmosomal proteins. This causes the entire epithelial layer, the epidermis, to detach from the basement membrane, resulting in severe, painful blistering.
And epidermolysis bullosa is similar.
Yes, it's an inherited condition resulting from genetic defects in these critical proteins, like the Integrins subunits or laminin 5 itself.
We shift now from how cells anchor to the environment to how they coordinate and connect with one another. Early experiments already proved that cells possess an innate ability to recognize and sort themselves.
That's the elegant, classic experiment summarized in Figure 7.20 here. If you dissociate embryonic cells, say ectoderm and mesoderm, and mix them randomly in a dish, they will first aggregate into a mass. But then they miraculously sort themselves out, so that like cells adhere only to like cells, eventually reforming the proper tissue structure.
This selective adhesion relies on a complex molecular code managed by 4 major families of adhesion molecules. Let's systematically review the 3 primary cell adhesion families, starting with selectins.
Selectins are integral-membrane glycoproteins that function as lectins, meaning they bind specifically to certain arrangements of sugars on other cells. This function is calcium-dependent.
And they mediate transient interactions, which sounds crucial for their main role.
It is. We have E-selectin on endothelial cells, P-selectin on platelets and endothelial cells, and L-selectin on leukocytes. They are perfectly designed for transient roles because they are optimized to form stronger bonds, specifically under the mechanical stress generated when cells are rolling along a blood vessel wall.
Next, we have the immunoglobulin superfamily, or IgSF. These proteins are defined by containing one or more Ig domains, making this domain one of the most abundant in the human genome.
While most IgSF proteins are involved in immune recognition, some, like VCAM, NCAM, and L1, mediate calcium-independent cell adhesion. This is a crucial distinction.
What's the major functional role of these molecules outside the immune system?
NCAM and L1 are absolutely vital for nervous system development. They guide nerve outgrowth and synapse formation. The clinical connection confirms this. Mutations in the L1 gene cause severe conditions, including fatal hydrocephalus or mental retardation, often linked to missing major nerve tracts.
Finally, the foundational molecule for tissue structure, cadherins.
Cadherins are large glycoproteins that mediate calcium-dependent adhesion and signaling. They define tissue boundaries because they typically engage in homotypic adhesion. E-cadherin on one cell only binds to E-cadherin on the opposing cell.
Structurally, how do they form this precise, stable connection?
Classical cadherins possess 5 tandem extracellular domains. The key is the calcium ion, which acts as a bridge, binding to and stabilizing the rigid conformation required for adhesion. They cluster together, forming a cell adhesion zipper. Just like Velcro, increased clustering drastically increases the overall adhesion strength.
And their role in tissue sculpting, or morphogenesis, is second to none, specifically through the process of epithelial-mesenchymal transition, or EMT.
EMT is a dynamic, critical process during embryonic development, essential for creating structures like the mesoderm and the neural tube. In EMT, stationary epithelial cells must lose their adhesion and become highly motile mesenchymal cells. The molecular mechanism is direct. This transition is defined by the loss or dramatic reduction of E-cadherin expression,
which lets them break free and move.
Exactly.
The sources move directly from these molecular principles to 2 crucial medical case studies that illustrate the dynamic nature of adhesion. Let's start with inflammation. Leukocyte recruitment. This is a beautifully choreographed molecular relay race.
It truly is a masterpiece of dynamic cell adhesion, a 5-step process that must happen quickly and accurately. Step 1, endothelial activation. Chemical signals from damaged tissue activate the nearby endothelial cells lining the blood vessel.
Step 2. Neutrophil trapping and rolling. What happens immediately after activation?
The activated endothelial cells quickly display P and E selectins. Circulating neutrophils bind transiently to these selectins, which dramatically slows their movement, causing them to roll along the vessel wall.
Okay, so that's the first slowdown.
Yes. Step 3. Neutrophil activation. Inside-out signaling. We've seen this mechanism before. The endothelial cell displays platelet activating factor, or PAF. This binds to a receptor on the neutrophil, sending an immediate signal inward, which activates the neutrophil's specific integrins, dramatically increasing their affinity.
Step 4. Firm adhesion. Now the neutrophil integrins are active, what do they bind to?
The highly activated integrins bind with high affinity to IgSF molecules, like ICOM1, also displayed on the endothelium. This immediate strong bond causes the neutrophil to stop rolling completely and adhere firmly to the vessel wall.
And finally, step 5. Invasion.
It changes shape dramatically, squeezes between adjacent endothelial cells, a process requiring the temporary disassembly of local adherins junctions, and enters the damaged tissue to begin fighting the infection.
If even one component of this elegant relay race is missing, the entire protective response fails. That brings us to leukocyte adhesion deficiency, or LAD.
LAD is harrowing. It's caused by a genetic lack of the beta-2 integrin subunit. Without this subunit, leukocytes cannot activate and bind firmly to the endothelium. They remain stuck, harmlessly circulating in the bloodstream. Patients suffer repeated life-threatening bacterial infections because their immune cells cannot exit the vasculature.
Shifting to cancer, the second case study is metastasis, which relies on subverting adhesion.
The initial steps of breaking free are rooted in the loss of adhesion, but penetrating barriers is accomplished by the mass deployment of those ECM-digesting matrix metalloproteinoses, MMPs. Cancer cells use projections called invadipodia, supported by MMPs, to literally push and degrade their way through the basement membrane.
We discussed the failure of MMP inhibitors, but what is the key molecular change that allows the cell to break free in the first place?
The key is the reversal of the process we saw in EMT. Malignant cells at the tumor edge undergo an EMT, characterized by the dramatic loss or reduction of E-cadherin levels. The data is unequivocal. Low E-cadherin correlates strongly with greater metastatic potential.
So loss of adhesion is a key step towards metastasis.
A critical one. Forcing malignant cells to express E-cadherin again dramatically suppresses tumor dispersal. It's so important that inherited mutations in the E-cadherin gene are directly linked to inherited susceptibility for stomach cancer.
Before we move on, the sources mention new technology for detecting these traveling cells.
Yes, new devices are capable of trapping rare circulating tumor cells, or CTCs, from a simple blood sample. This allows clinicians to analyze the molecular characteristics of the traveling tumor cells, including their E-cadherin status, to monitor treatment progress and predict the likelihood of dispersal.
Let's detail the specialized junctions where these adhesion molecules aggregate, typically forming a junctional complex near the apical surface of epithelia. First, adherens junctions.
These form belt-like structures that completely encircle the cell, tightly binding it to all its immediate neighbors near the apical surface.
How does the internal structure of the adherens junction link to the cytoskeleton?
Cadherins bridge the narrow external gap. Internally, the cytoplasmic domain is linked via adapter proteins like alpha and beta catenins directly to the actin filaments of the cytoskeleton. This mechanical linkage also serves as a critical signaling hub.
You mentioned beta-catenin as crucial here. What is its signaling role?
Beta-catenin is a key element in the WANT signaling pathway, which is essential for developmental patterning and gene expression. So the adherens junction acts as both a physical anchor and a signaling sensor.
Second, we have desmosomes. These are disc-shaped patches, not belts, but they are essential for high-stress tissues, like cardiac muscle and skin.
The cadherins here are specialized as desmogleins and desmocollins. Crucially, the dense cytoplasmic plaques anchor large bundles of intermediate filaments, keratin, which loop through the plaques.
Ah, so keratin again for strength.
Exactly. This intermediate filament network provides tremendous, stable structural continuity and tensile strength to the entire cellular sheet, distributing tension across many cells.
And just like with hemidesmosomes, targeting these structures leads to severe blistering disorders.
Correct. Pemphigus vulgaris is an autoimmune disease where antibodies attack a desmoglein, causing the loss of epidermal cell adhesion. Because the cells are no longer stuck to each other laterally, the epidermis detaches and results in painful blistering.
This entire section reinforces the concept of transmembrane signaling. Every molecule we've discussed, integrins, cadherins, selectins, and IgSF, has the potential to transmit signals across the membrane.
Yes. By linking to the cytoskeleton and regulatory molecules, the outside signal is translated inward, altering cell behaviors like growth, motility, or survival. We circle back to the mammary gland example from figure 7.29. Epithelial cells flatten on a bare dish, but when laminin is added, binding to their integrins, it activates internal kinases. This outside-in signal restores the cell's differentiated state.
Beyond adhesion, cells and epithelia need to create a leak-proof seal. This is the domain of tight junctions, located at the very apical end of the junctional complex.
Their function is twofold. First, they act as a barrier, strictly preventing the diffusion of water and solutes through the paracellular pathway, the space between adjacent cells.
Okay.
Second, they act as molecular fences within the plasma membrane, blocking the diffusion of integral membrane proteins between the apical and basal domains, which is essential for maintaining the cell's polarity.
How does this structure achieve this leak-proof seal?
The key structure is revealed via freeze fracture analysis. It shows interconnected strands running parallel to the apical surface that completely encircle the cell like a gasket. The quality of the seal is directly related to the number of these parallel strands present.
And the molecular basis of that permeability. We originally thought it was one protein, a cludin, but the sources clarified the true structural components.
Right. The major structural component is the family of proteins called collagens. There are at least 24 types, and these proteins form the core of the TJ strands. They are critical because they determine the selective ion and solute permeability of the junction.
The sources provide two striking clinical examples that illustrate both the selectivity and the sheer necessity of claudins. Let's start with claudin 16.
Claudin 16 is specifically expressed in the kidney tubule. Its job is to form pores that are permeable to magnesium ions, allowing for magnesium reabsorption. Patients with mutations in the claudin 16 gene suffer from low blood magnesium levels because the ion simply cannot be reabsorbed through the TJ and is excreted in the urine. This highlights the selective transport function of a TJ.
And claudin 1 highlights the crucial barrier function.
Yes. Mice engineered to lack claudin 1 die shortly after birth from dehydration. Why? Because they cannot assemble watertight TJs in the outer layers of the epidermis, the skin. This dramatically demonstrates that our ability to retain water depends entirely on these microscopic molecular seals.
Tight junctions are also the foundation of the legendary blood-brain barrier.
The TJs between the brain capillary endothelial cells are exceptionally tight, among the tightest in the body, preventing nearly all substances from passing from the bloodstream into the delicate brain tissue. This protects the brain from toxins, but it also poses a massive pharmaceutical challenge, finding a way to temporarily and safely open these TJs to deliver vital therapeutic drugs.
Now we move to specialized sites that facilitate direct communication between the cytoplasm of neighboring cells, starting with gap junctions in animal cells.
Gap junctions are defined by a tiny 3-nanometer cleft separating the plasma membranes, spanned by precise molecular pipelines. They are essential for chemical and electrical coupling.
How are these channels constructed?
They are built from the integral membrane protein connexin. 6 connexin subunits assemble into a ring structure called a connexin, which spans the membrane of one cell. Connexins from opposing cells align perfectly to form a complete hydrophilic channel, or annulus.
This channel size limits what can pass through, what molecules are exchanged.
They allow the non-selective diffusion of small molecules, up to about 1,000 Daltons, things like ions, ATP, cyclic AMP, and sugar phosphates. Critically, these channels are gated. They can be opened or closed, primarily triggered by connexin phosphorylation, voltage changes, or significantly abnormally high cytosolic calcium concentrations, a protective mechanism to isolate a dying cell.
This rapid exchange enables two key physiological roles. First, the electrical synapse.
In tissues requiring synchronous action, like cardiac muscle, gap junctions allow the immediate flow of ions, facilitating the rapid spread of electrical impulses from the sinoatrial node, enabling the heart chambers to contract simultaneously.
And second, metabolic cooperation.
In avascular tissues, like the lens of the eye, cells deep within the structure lack a direct blood supply. They rely on gap junctions to share essential metabolites and nutrients with cells closer to the source.
Mutations in connexin genes are clearly linked to human disease, including deafness, blindness, and cardiac arrhythmias. On a different note, the sources also describe the newly discovered tunneling nanotubes.
These are a new form of long-distance communication in cultured cells. Thin, elongated tubules supported by actin filaments. They are capable of transporting much larger cargo, including proteins, vesicles, and even viral particles over long distances between cells.
Moving to plants, they have a communication problem due to the rigid cell wall. They solve this with plasmodesmata.
Plasmodesmata are cytoplasmic channels that literally punch through the thick cell walls of adjacent cells. The channel is lined by the plasma membrane and usually contains a dense central structure called the desmotubule, derived from the smooth ER. Substances pass through the surrounding annulus.
We used to think these were highly restrictive pores. Has that understanding changed?
Yes, significantly. While they were thought to exclude molecules larger than 1 kiloday, we now know the pore is capable of dilation, allowing the passage of much larger molecules up to 50 kiloday. Plant viruses exploit this by encoding movement proteins to increase the pore diameter.
Plants also use their own equivalent of movement proteins for internal signaling.
Absolutely. They regulate the flow of their own proteins and RNAs through the plasmodesmata, which is essential for integrating plant-wide activities. Figure 7.35c illustrates this beautifully, showing a transcription factor moving from one cell layer to the next to regulate gene expression.
Our final major structural topic is the cell wall, the protective outer envelope found in plants, fungi, and some protists. It serves as the plant's structural foundation.
It performs multiple critical functions, providing mechanical support, maintaining the cell's fixed shape by resisting immense internal turgor pressure, protecting against abrasion and pathogens, and mediating cell signaling.
We can understand the cell wall by thinking of it using the structural analogy. Reinforced concrete or fiberglass. It has a fibrous element embedded in a gel-like matrix.
The fibrous element is the cellulose microfibril. These are rod-like bundles composed of roughly 36 parallel cellulose molecules held tightly by hydrogen bonds. This component confers maximal rigidity and resistance to tensile or pulling forces.
And the surrounding gel-like matrix is composed of 3 key macromolecules.
First, hemicelluloses. These are branched polysaccharides that bind directly to the cellulose microfibrils, effectively cross-linking them into a single resilient structural network.
Second are the pectins.
Pectins are negatively charged polysaccharides. Like animal proteoglycans, their negative charge allows them to hold large amounts of water, forming an extensive hydrated gel that fills the spaces between the fibers. And yes, they are used commercially to give jams and jellies their gel-like consistency.
The matrix also contains important proteins, like the intriguing expansins.
Expansins facilitate cell growth. They are responsible for causing localized relaxation of the cell wall structure, allowing the cell to elongate in response to the internal turgor pressure. The wall also contains protein kinases that transmit signals sensed externally inward to the cytoplasm.
The synthesis of these components is spatially separated, highlighting the complexity of building this exterior structure.
It is complex. Cellulose is polymerized at the cell surface by the enzyme cellulose synthase. This enzyme is organized into a 6-membered ring, or rosette, embedded directly in the plasma membrane. Crucially, the direction of movement of this rosette, which lays down the microfibrils, is precisely guided by cortical microtubules located just inside the cell boundary.
So the internal cytoskeleton dictates the structure of the external wall.
Precisely. In contrast, the matrix components, hemicelluloses and pectins, are synthesized internally within the cytoplasm, primarily in the Golgi complex, packaged into vesicles, and then secreted out to the wall.
Finally, let's briefly distinguish between the walls found in young versus mature cells.
We have primary walls in young, actively growing cells. These walls possess extensibility necessary for cell enlargement. When the cell reaches maturity, it often forms a secondary wall, a thicker, more rigid layer containing increased cellulose content and incorporating lignin, a complex polymer that provides maximal structural support, forming the major component of wood.
What a detailed tour. We've gone from the immediate sugar shield of the glycocalyx all the way out to the rock-solid structure of the plant cell wall.
The essential lesson of this deep dive is the breathtaking interconnectedness of structure, adhesion, sealing, and communication. Whether we are discussing collagen providing the frame or tight junctions providing the seal, everything relies on highly specific, targeted molecular handshakes.
And at the heart of the regulatory systems are those cell surface receptors, especially the integrins. They aren't just anchors, they are signal processors, constantly translating the chemical state and the physical state of the outside world into signals that govern whether a cell grows, moves, survives, or differentiates.
It forces us to confront the true meaning of multicellularity. An organism is held together by trillions of these instantaneous, specific molecular handshakes happening every single second.
Hmm.
And if we consider the continuous dynamic tug-of-war, the tension generated by integrins pulling on the matrix, counterbalanced by the constant remodeling action of MMPs, doesn't the regulation of that dynamic equilibrium fundamentally define tissue health, disease vulnerability, and the entire process of aging?
It suggests that tissue health is less about static structure and more about maintaining a perfectly balanced dynamic tension.
That is the essential takeaway. The outside world is the ultimate dynamic regulator of the cell.
Thank you for letting us guide your understanding on this deep dive. This has been your essential recap from the Last Minute Lecture team.